Good morning. My name is Julika, and I'm a postdoc at the Julika lab at Stanford, working in the Craniofacial research group. And today I'm presenting to you a study characterizing the mouse model of Frank-ter-Haar syndrome. Frank-ter-Haar syndrome is a rare genetic disease that is characterized by craniofacial dysmorphology and skeletal malformations. This is due to mutations in the SH3-PXD2B genes, which encodes for the proteasomal adapter protein TKS4. TKS4 is important for the formation of proteasomes, and proteasomes are actin-rich cell productions that degrade or help degrade the extracellular matrix and thus promote cell migration. So in this study, we analyze a mouse model of Frank-ter-Haar syndrome. For breeding, we utilized heterozygous mice and then harvested the skulls of homozygous mice to process for histology, micro-CT, and immunofluorescence. We harvested dura mater cells and osteoblasts from these skulls to process for in vitro assays for osteogenic differentiation, proliferation, migration scratch, and immunocytochemistry. In vivo, we performed cranial defect surgery using a 2 mm trephine drill to assess bone regeneration and migration. Moving on to the results, in figure 1a, we can see a photo of a mutant mouse in the front and a wild-type mouse in the back. The mutant mouse shows a generally smaller skeleton and a shortened frontal bones and a domed skull. Figure 1b shows micro-CT analysis of skulls of wild-type and mutant mouse on postnatal day 30. We can see that in the mutant mouse, there's hypermineralization both in the frontal and the parietal bone, almost resembling foramina. Both the posterior frontal and the sagittal suture is enlarged and patterned. We then moved on with histological analysis, and we performed pentachrome stain on histological sections of the PF suture, which you can see in figure 2a, and the SAT suture in figure 2b. The PF suture has been analyzed during the time of endochondral ossification, so during the time of physiological fusion on postnatal day 12, and we can see cartilage formation in the wild-type mouse. We can also distinguish two bone layers, so an endocranial and an ectocranial bone layer. In a mutant mouse on the right side, we can only see one bone layer, and in the middle there's membranous suture mesenchyme and no cartilage formation. So the PF suture does not undergo endochondral ossification. Then in figure 2b, we can see the SAT suture on postnatal day 30. At this time, it's physiologically patterned in the wild-type mouse. And then in the mutant mouse on the right side, we can only see the suture mesenchyme in the respective area. No bone fronts can be distinguished. And then we also see hypermineralization, and we see ectopic bone formation in the later time points. Moving on to the in vitro assays, we can see here a migration scratch assay in figure 3a with using dura mater cells, and the left side the wild type, on the right side the mutant mouse. We can see that after 24 hours, the wild type dura mater cells have migrated into the scratch area, whereas in the mutant mouse, we can see almost no migration. We confirmed this in vivo assay, so we performed a 2 mm per vial defect in the parietal bone, and we harvested the skulls 24 hours and 48 hours post-surgery and stained for H&E. So on the left panels, we can see that the defect area, and we can see that there's cell migrating into the defect areas, whereas on the right side, we can see the mutant mouse, we can only see the dura mater. There is no migrating cells into the defect area. We then performed immunofluorescence on dura mater cells. We stained for the proteasomal marker protein cortactin in green, and for the cytoskeletal marker phalloidin in red. Upon TGF-beta induction, we can see that there's proteasome formation in the wild-type dura mater cells, but no proteasome formation in the mutant cells. So we conclude that this mouse model is characterized by severe craniofacial dysmorphology and disrupted suture patterning. The DM cell migration and the proteasome formation is impaired both in vitro and in vivo in the mutated mouse. So we conclude that this mouse model represents a valuable model to study Frank-ter-Haar syndrome and to unveil novel molecular mechanisms that lead to this disease. In the future, we'd like to move on with transcriptomic and proteomic analysis of mutant and wild-type mouse skulls. We'd also intend to create a triple or two triple mutant mouse lines using Wnt-Cre and MESP-Cre, cross it with a MTMG double fluorescent reporter mouse and the SH3-PXD2B mouse to allow for neurocrest cell and mesodermal cell lineage tracking during skull development. We thank you very much for attention, and if you have any further questions, I'm happy to answer via email, which you can see on the bottom left side of the screen. Thank you very much for listening.